Kate Scow here. This is Soil Microbiology, and this is Lecture 8. And Lecture 8 is about the rhizosphere, and we'll feature mycorrhizal fungi as part of that. I'm also going to ask you to read a short review article that will cover this material as well as the nitrogen fixation lecture. So here's a picture showing the plant root as it really is, surrounded by this rich world that is filled with microbes that provide all these benefits to the plants, and of course the plants are giving the microbes something back. This is a terrible, impoverished world in the absence of a rhizosphere, which isn't what you're going to find very often. So we're going to talk about the rhizosphere itself, particular conditions of it. Why is it different from what we call the bulk soil, which is the soil that is not under the direct influence of plants. And then we're going to focus, as I said, on mycorrhizal fungi, particularly on endos and ectomycorrhizal fungi. So just want to start out letting you think about plant roots. There is no one kind of plant root system. These special ecosystems that are immediately around the roots of plants that we call rhizosphere is going to vary substantially. And these are just grassland plants, but with trees you can imagine different kinds of arrays, even more so. And there's going to be differences in the density of the roots, the branching patterns, how deep they go, and chemical composition, many other aspects. And then if you go down and drill down on what's going on immediately around the root, here is a crazy close-up of this sort of complexity and, as you might be able to infer, this frenzy of biological activity in the immediate vicinity of the root. So when you look overview at plant root microbe interactions, there's a variety of different things that go on. By the way, some of them go on above ground. We're not going to talk so much about the epiphyte or the phylloplane, which is the area on the surface of the roots that also has microbes. We're focusing below ground, and there's both beneficial as well as negative interactions going on. Let's just start with the negative. Obviously, there's a lot of plant pathogens down there as well. There's also organisms that are going to be grazing plant roots even while they're living, and stealing resources in a way from the plant. But there's a lot of beneficial interactions that are going to really help the plant to survive. In fact, the plant would not survive without all of this additional help. Nobody is alone, nobody is an individual, and you can almost think of the rhizosphere as the stomach of the plant. The stomach is without a particular boundary like our stomach has. It's actually sitting right smack in the soil, but it's a place where a lot of the things the plant needs to do to be able to survive is going to go on. So this includes plant growth promoting bacteria and fungi. This is this term PGP R that you see sometimes in the literature, these plant growth promoting organisms, as well as the nitrogen fixers and mycorrhizae, some symbiotic relationships, some less so. So that just gives you an overview. Let's look at defining the rhizosphere. It's the zone of soil immediately adjacent to and under the influence of the plant root. So you can imagine how you are thinking about what particular aspect of the plant is involved is going to influence this area that's called the rhizosphere. Some attributes of the plant root are going to be very, very close to the root, and some attributes are going to extend further out in the soil. Operationally, we define the rhizosphere as the soil that clings to the plant roots after gentle shaking of a certain time period. It's a kind of a crazy definition that is very difficult in a way to replicate it, but it's a way that people do studies where they compare rhizosphere versus what we call the bulk soil, the area that is away from the immediate influence of the plant roots. So in the rhizosphere, we see higher carbon. We'll talk about where that comes from in a minute. Higher availability of carbon forms, Different pHs, it can be either higher or lower depending on whether or not acidification reactions are occurring or reactions that consume protons. You can have higher or lower ion concentrations because certain ions are going to be sucked up by plants, making depletion zones immediately around the root, and others are going to be excluded by the plant, even though they may be drawn through transpiration around the plant root, but the plant might not take them up, so they can concentrate there. There's going to be greater compaction of the soil immediately around plant roots because it's actually pushing up against the soil. But on the other hand, on a bigger scale, and as plant roots decay, there's going to be increased porosity that is a result of those plant roots having channeled through the soil. We have microbial communities that are different in the rhizosphere than the bulk soil, although it's pretty hard to make big generalizations about it. There's a lot of studies about it, but it's pretty complicated. And greater fluctuations in soil water potential immediately adjacent, immediately in the rhizosphere. Okay, let's look at that carbon. 30 to 60% of the carbon fixed by plants through photosynthesis is released through the roots. People used to think this is crazy, this is terrible, how can we stop this? What can we do to breed the plants so they don't leak all their carbon out? This is really a serious issue. What are we going to do? We're not getting as much yield. But now people know that plant root, the carbon is actually getting some benefit when it is released in the rhizosphere. So there's different ways of classifying these different forms of carbon that come out of the roots. But we can divide it up into exudates, which are these low molecular weight compounds shown by the number ones that are just going to be diffuse out or be transported out from plant roots, and they could be things like sugars, amino acids, nucleic acids, exudates, and secretions, the only difference being whether or not there's a transport or diffusion involved. And then we have this more complex stuff, which is mucilages, and that is higher molecular weight material, cellulose, pectin, lignin, that's like sloughing off from the plant roots as it grows through the soil. You can imagine that the soil is going to kind of like scrape off some of the material from the plant root as it grows. So that's going to be slightly more complicated, the breakdown of that, like the carbon cycle that we just looked at. And then musagel, this magical stuff, which is a combination of plant exudates that cells microbes and their extracellular polymers and soil minerals. And these musagels are really important and fill the gap between the plant root and the soil around it, and really are important for keeping the plant root in touch, in contact with the soil as it dries out. It won't make a gap between it as easily if it's filled with this musagel material. And then there's also lysates, which I think is less important here, but which is from plant cells just exploding and releasing material in it. So a lot of different kinds of exudates, and the rhizoplane, by the way, is a definition of the surface of the plant root, which is where some microorganisms are associated. Okay, so life for microbes in the rhizosphere. If you look at microbes there, you can see their spatial distribution is pretty patchy. Only 7 to 15% of the root surface is covered, so not all root surface is equal. For example, the root cap, which is going to be here, at the growing tip of the root, is generally sterile. Very few organisms, but right like here in these areas immediately adjacent, there's a lot of, as I said, sloughed off cells, and there's a lot of microbial colonization there. Also, where there are any holes or crevices or where root hairs connect, there's going to be microbes that are going to be there. And associated with different places where carbon is released, there's going to also be greater densities of organisms. Microbes in the rhizosphere tend to be bigger and fatter, larger sizes than in the bulk soil, and containing more storage compounds like polyhydroxybutyrate, PHB, which is kind of like a microbial fat. So rhizosphere organisms seem to have more of that. They have higher growth rates in general than organisms from the bulk soil, and a lot of them are attached. They're going to be attached to the plant root through polysaccharides or fimbriae, which are little like flagellum-like stalks that actually hook them up to the plant root. It's very, very competitive in the rhizosphere. There's a lot of organisms there, and they are all duking it out for that carbon. So it's pretty difficult, actually, to inoculate outside organisms into these environments where there's a lot of local organisms that are pretty well adapted that are going to compete for the resources that the inoculant is trying to get. Although sometimes if plants are inoculated really early on, particularly trichoderma is a fungus that is used to inoculate seeds or seedlings in greenhouses before you put them out into the soil. And if the plant root is already colonized by this trichoderma when it first hits the soil, trichoderma is neutral, doesn't do anything bad to the plant, and it can actually block out colonization by some of the other organisms. Sometimes there's a lot of interest in that as a kind of biocontrol, that's sort of inoculation. Inoculation in the sense of like a vaccine, vaccinated, kind of. All right, so here's just an overview of the rhizosphere, and you think about the three players: the plant, the soil, and the microbes. So the plant has certain characteristics that are going to interact with the soil, and it has a lot of activity, including this exudation, but it's also going to be growing roots all the time, extending into new areas of soil, and it's going to be taking up nutrients, and that's going to have impact on microbial activity as well. All the things that we've already learned about in this class, you can connect up to what's going to happen in the rhizosphere. And there, the activities of the organism, their total biomass, their biodiversity is very much going to influence then how they relate to what the plant does, and all of these different traits are interacting with one another. So how does the rhizosphere organisms influence plants? They change the soil chemical composition. Obviously, this is total Soil Science 111. Review: mineralizing organic matter, producing acids that are going to dissolve, minerals that are nutrients, or sources of nutrients for plants like phosphorus. They themselves are going to turn over. Remember, they are a pool of the soil organic matter, and they are going to be giving themselves up for the plants when they die. Phase equilibria of nutrients are going to change as a function of what microbes are doing to pH and redox, and also in the complexes that they're going to form, which we'll learn a little bit more about in the next lecture. But that's going to very much influence the regime of nutrients around the plants, and also soil structure. There it is again. They can change morphologically and physiologically some of the root properties by producing these plant growth regulators, things like auxins, gibberellic acid, ethylene, which are chemicals that regulate what plants are going to do in response. And it's really part of this big, rich conversation that's going on in the soil between plants and microbes in either developing symbiotic relationships, for example, with the mycorrhizal fungi, and also the nitrogen-fixing rhizobia. There's a lot of back and forth that we'll talk a little bit in tomorrow. Not tomorrow, but in Tuesday in the next lab, where we're going to talk about nitrogen fixation in particular. There's, as I said, a lot of signaling that's going on that happens via these production of these chemicals that the organisms send back and forth to each other and alter. Also, in symbiotic relationships, rhizosphere organisms influence plants by directly transferring nutrients into the host, nitrogen, phosphorus, in return for something back. And also, microbes can block root surfaces and compete for nutrients. And sometimes you see this with phosphorus and some of the other ions. And it may also depend on, and we know about immobilization of nitrogen, but this can be temporary and may actually benefit the system by keeping things around. Pathogen suppression is another benefit that's provided by rhizosphere organisms. And then we talked We just talked about mucigel that's really important in drought tolerance of plants. That's this complex mix of microbes, minerals, and plant exudates. Okay, so we're going to talk about a very special relationship, the mycorrhizal fungi, which is the symbiotic association between plant roots and fungi. This extremely common relationship. Most plants have mycorrhizal fungi. Very few plants do not have mycorrhizal fungi. Can increase the plant root length 100-fold or even more through these extensions that are actually these mycorrhizal hyphae, or this mycelia that's connected up to the plant roots but goes out into the soil. And you can see this picture isn't really to scale, but you're looking at this sort of extension of this hyphal network. There's a mushroom popping up out of this hyphae that's associated with this particular tree. So it increases the nutrient foraging abilities for the plant, and the fungus gets carbon back. There's a variety of different kinds of mycorrhizal associations. I think there's like 5 or 6 kinds, but we're going to focus on 2 kinds, the ectomycorrhizal fungi and the endomycorrhizal fungi, which used to be called vesicular arbuscular mycorrhizae, or VAM. So you're going to sometimes see that name. The ectos are associated with gymnosperms and angiosperms, so they're generally woody plants. So these are the ones that you're going to see in trees, the ectos. And the fungi are basidiomycetes and some ascomycetes for the most part. They have some characteristic structures that we'll look at in the next few slides, and they are important in helping the plants take up nutrients, but also with aggregation. We're going to look at these functions in later slides. In contrast to the endomycorrhizal fungi, so they're associated with non-woody plants. Virtually all the herbaceous plants that you see out there in the world, it's almost every plant has mycorrhizal associations. There's a very few sort of weedy plant families that do not have mycorrhizae, like the brassica don't, but most do. It's very rare not to have them. And the fungi involved tend to be in the zygomycetes, and this glomalis is a very famous one. You may have heard the term glomalin, which is the extracellular polymeric material that's released by fungi, glomalin, that has its own sort of speckled history. It's not as clear-cut as people used to think it was, but that's associated with this organism, this endomycorrhizal fungus. Okay, so let's look at the ecto first. The ecto is the one that you can see with your eyes, and in fact you can pull up a plant root and see this rich sort of sheath of hyphae around it. If you blow that up, here's the plant root, and you can see that growing around it is this coating almost of these hyphae that create what we call a mantle or sheath surrounding the roots. And here's a cross section of the root showing the cortical cells, and then this big mass of hyphae around it. So the Hartignet is the inner part of that sheath and includes the hyphae growing in between that outer, coating and the plant root. So it's this sort of material or area section in here. And here's a close-up looking at these hyphae that are growing a little bit into the plant root, but they don't go that far in. They tend to be associated just with the cortex and on the outside, and as I said, you can see it. So the exchange of nutrients occurs in the Hartignet. That's the place where there's the back and forth of what the plants give the fungi and what the fungi give back to the plants. So as I said, you can see them. Here are non-mycorrhizal plants, or plants that were deprived of their mycorrhizal associates, and here's how it looks with them. Likewise, I think this might be a pine tree root without and with. You can totally see it with your eyes. And that's just a beautiful picture, cartoon showing these mushrooms coming up And you do see these particular mushrooms that are associated with the ectomycorrhizal fungi that have colonized trees and forests. So a lot of those mushrooms, those classic toadstools, are actually the fruiting bodies of ectomycorrhizal fungi. Pretty cool, huh? Okay, in contrast, the endomycorrhizae fungi here or arbuscular mycorrhizal fungi are not very visible with your eye. They don't form as thick as sheaths on the outside of the plant roots, and they're called endo because they penetrate more inside the plant roots into in between the cortical cells, and also pushing into it. I don't know that they actually go inside of it. They may still have a membrane separating them, but they're moving in there, and they have these characteristic organs or organelles. One are the arbuscules. That name arbuscule comes from that. Little trees, and that's the area where the nutrients are transported back and forth between the plant and the fungus. And sometimes there's these vesicles as well that I don't know if people have a good idea what they actually are. And here's a stained slice of that showing these arbuscules here that are, pushing inside the plant cortical cells. So, the vesicles may be storage compounds. And here's a picture showing, another rendition showing that you have these hyphae that are growing out in the soil from this colonized area that has more presence inside the plant root. But of course, these hyphae extend far out in the soil for great distances. And just in case you didn't pick it up or I didn't make that clear enough, also here, even though you have this like sheath around it, there's also lots of hyphae extending far out into the soil looking for nutrients that are going to be transported back into the plant. So that's a trait of both. So the benefits of the plant, I like to call it a cheap root. It increases the soil volume that the root can exploit through this relationship with the mycorrhizal fungi. It's a relationship they can shut down at any time, so it's not as much of an investment of the plant's own resources into making more roots. So it's just a much more efficient way of getting out into the soil, and also particularly the benefit of using fungi is they're going to be able to access smaller pores than plant roots are going to be able to get into. So a benefit of that is phosphorus uptake. Phosphate is very immobile, very insoluble. It is not going to move to the plant with transpiration during photosynthesis, unlike nitrate, which is very mobile. Phosphorus, the plant has to go after and go get it, hunt it down. So mycorrhizal fungi are really good about that. So effectively, they decrease the diffusion path length by going directly to the P sources, moving into these small pores, and they can take up that phosphorus and just translocate it back through the hyphae without any kind of interference with the soil chemistry that might want to make that phosphate fall back out of solution. Also, a lot of mycorrhizae have phosphatases, so enzymes that are good at getting access to organic P, as well as may be good at dissolving mineral forms of phosphorus as well through compounds like oxalic acid, which a lot of forest fungi have that are good at dissolving rock phosphorus or mineral forms of phosphorus into phosphate. Okay, ectos also, and I don't know, I think increasingly endos also are recognized that they could be important in nitrogen uptake, important for resisting drought periods, and particularly the ectos can, by just creating that physical sheath around the plant roots, can protect them from heavy metals and perhaps also pathogens. So there's a lot of benefits to the plant. And the benefits to the fungus is carbon, and so up to 20% of the plant carbon may go directly to mycorrhizae, remember the Arctic net in the ecto, or the arbuscules in the endos, or arbuscular mycorrhizal fungi is the place where this exchange occurs. So these are going to be different kinds of sugar compounds, and they also may actually provide growth factors to the fungus as well that may come directly from the plant. There's some really cool ideas and research on are those mycorrhizal fungi sort of networks below ground, are they actually letting plants physically connect to one another? And are plants communicating to one another below ground through these interconnected mycorrhizal networks? And are there nurse plants, like older plants, that can help nurture young plants that are just starting up through feeding them through their mycorrhizal fungal networks? So lots of really sort of nice ideas, exciting hypotheses that people study sometimes by putting chemicals into one plant, or nutrients that are labeled in some way that they can tag and then see if they're taken up into an adjacent plant, and whether or not that relationship does exist. And I sent you to the via the Smart site a link to a short YouTube video about this in forests. So pretty exciting stuff. And then soil structure, you know how much I love soil structure. I can never resist. And the rhizosphere and mycorrhizal fungi are a really important area for aggregate formation. And here we're talking more sort of at this scale. We have the extracellular polymers maybe being more important at this scale, although there's also important at this scale. And so this is like the 0.3 millimeter to 3 millimeter scale, where they're going to be very, very influential in creating these larger aggregates that are so important for the soil. Okay, that's it. Short and sweet, huh? We're over. Okay.